vaccines, but also a little bit about got lost here, a little bit about the sociological and other aspects of vaccination. And it's kind of fun to go back and look at that. First thing I should say is that I had already planned to talk only about these, but I wanted to talk a little bit about vaccine failures because I think they're just as important. And unfortunately, I don't think I have enough time to do that, except for one a small example. But these are the things that I'm going to talk about because each of them represent an incremental understanding of how vaccines get made, and starting with smallpox, then yellow fever, polio, hepatitis B, a little bit about adjuvants, and finally about the RNA vaccines, of which I suspect SARS-CoV-2 is only the beginning, and there will be many more. And with a little bit of luck, we can get through all of this. And so the concept that you could infect someone and then prevent future infection, I'm sure was already there. People recognize that you only get smallpox once. But the idea of looking at variolation or the deliberate infection of individuals with the smallpox virus really started in China. And this is a figure that came out of some Chinese manuscript. Obviously, there's something wrong with it in that it says figures showing vaccination pustules, when in fact vaccination as a word had not really been used for quite some time later. But from China, this idea that you could take pustules, particularly someone who had variola minor or the less fatal or less virulent form of smallpox, and inoculate someone, and that they would then not be likely to get the real disease, spread out into India and then to the Ottoman Empire. And it was there that this lady, Mary Montagu, who was the wife of the British ambassador, learned about variolation, actually variolated her children, one of them in Turkey and one in the UK, somewhere about the beginning of the 18th century, and started spreading the word that this was a great way to prevent smallpox. She was a very wealthy lady, as would have been expected, as the wife of an ambassador, had a whole big, nice pile up in York. And this obelisk, I believe, is there, and it was placed by one of her friends in memory of her and of the contributions that she made to England in terms of the decrease in smallpox. So variolation actually became quite common there. And it did have a little bit of a mortality. Occasionally it would spread, but it was considered good enough, at least for that time, since the mortality of smallpox was quite high. And there were lots of people who were invested in the idea of using variolation as a prevention for smallpox. On this side of the Atlantic, it wasn't as popular, although Benjamin Franklin lost his son, his first legitimate son, so to speak, Frankie, to smallpox, and he became a big advocate of variolation. And so he was such an advocate that he had a friend write this pamphlet, of which we can see the title page, which gave plain instructions on how to do this, and had a foreword written by him. And at this point, sort of the middle of the 18th century, Benjamin Franklin was extraordinarily well known throughout the world, throughout Europe and the United States, as many of you can, looking at this recent PBS series, can see. And they briefly mentioned the fact that he lost his smallpox and became a big advocate of this issue. Now, the Continental Army was particularly susceptible to smallpox. The British soldiers usually either had smallpox as children or they were variolated, and so they were immunized. And the British knew this, and there was some suspicion that they would actually try to deliberately infect the revolutionary forces. They did that either by graving blankets and maybe adding some sex workers who were already infected, and they turned to follow armies. And so the father of our country became quite concerned, and he actually said, well, we need to variolate or inoculate everybody who's in the army. And this is a letter that he wrote to John Hancock, who was then the president of the Continental Congress, or the sort of president of the country, if you will, in February of 1777. And you can see the part that I have highlighted in red, that it says, I have therefore determined that not only to inoculate all the troops now here that have not had it, but shall order Doc Shippen to inoculate the recruits as fast as they come into Philadelphia, proving 2 or 3 points. First of all, that the mandates for inoculation didn't just start last year, and that the fathers of our country, the elders, were in fact very much in favor of this. Now you ask yourself, why in Philadelphia? Well, that happened to be where all the army was congregating at that point. Those of you who know history, this type of history, know that in September of 1777, they had to escape Philadelphia and move to Valley Forge because the British were, in fact, captured Philadelphia and kept it for most of the war after that. Dr. Shippen, who was listed here, again, another one of these august people of the 18th century, was a third Surgeon General. I'm not sure exactly what happened to the first two, but he was from Philadelphia. He was one of the founding faculty members at the University of Pennsylvania, and he had gotten his degree from the University of St. Andrews, where he had been sent by Benjamin Franklin and some of the other big shots from Philadelphia to learn medicine together with John Morgan and one other person, and come back and create the first medical school in the country. So he was in charge of inoculating the recruits in Philadelphia. I'm not sure what he did. Once they went to Valley Forge, they may have had a different Surgeon General. So the variola, which is smallpox, can be divided as a disease into variola major and variola minor. And the minor is really the milder forms, it's just cutaneous, and the major is the one that is quite fatal. But it's important to recognize a couple of things here. First of all, it's a group of viruses. Cowpox is the original vaccinia. Vaccinia is the strains that are still available for contemporary use. And then there's monkeypox. And monkeypox can infect humans, although it does not spread as effectively as smallpox. And Jenner, of course, is always credited with the first vaccine. He practiced country medicine after being an apprentice. He was eventually granted an MD, and that is the correct degree, because at that point, the University of St. Andrews was giving an MD, not the whatever they do now, which is more complicated. And his first observation was that people who had had the cowpox could not be variolated. So he dutifully wrote a paper about this, and the people who were reviewing the paper immediately rejected it. He was probably not of the right academic type. And so then he went back and said, well, let me do some cowpox inoculation, and I'll tell you about that story in a second. And that's when eventually that was published. And this little hut here, called the Temple of Vaccinia, is the place in his estate near Berkeley in the United Kingdom, where he did most of his vaccination, and which is kept now, obviously, as a temple to vaccine. Now, this is his first description, and I'm going to read most of it here, at least the parts in red. And it says, to more accurately observe the progress of infection, I selected a healthy boy about 8 years old for the purpose of inoculation for the cowpox. The matter was taken from a sore on the hand of a dairymaid, Sarah Nelms, and here's a drawing of her hand, and it was inserted on the 14th of May, 1796, into the arm of the boy by means of 2 superficial incisions, barely penetrating the cutis, about ½ an inch long. And so the boy was fine, and so he decided he was going to inoculate him now, some 6 weeks later, with a variolous matter immediately taken from a pustule to see if he would get infected. He did not get any disease, and so several months afterwards, and these are obviously not IRB approved experiments, he was again inoculated with variolous matter, and no sensible effect was produced on the constitution. And having done this a few times is when he decided he would go ahead and write a paper about it. And this is from one of his drawings, the cowpox sores on the hand of Sarah Nelms. Now, it's good that they remember the milkmaid, as they were called at the time, who gave her hands to such an important effort. But the British have a tremendous sense of history, and I'll show you what else they did. Jenner, in fact, was awarded a lot of prizes, totaling up several $1 million in today's currency. He was never knighted, which is a real slight, and he continued vaccinating in his home in Berkeley. Now, a lot of people were invested in variolation, and so they were very resistant to the idea that he was doing this with cow. There was a whole medical enterprise, just as there is now, based on the particular procedure. And so this cartoon from the Morgan Library kind of illustrates what the feeling that some people had, which was that, in fact, that is mine, I better get rid of it. Here is an individual being inoculated with vaccinia, and you can see all these people, and here's vaccinia poc, which is being taken from, who are sprouting cows in all kinds of places, including their nose, their ears. This lady is already a full cow coming out of their mouth. And this is really the wonderful effects of the new inoculation, via the publications of the Anti-Vaccine Society, no different than what we have now. And if you look carefully, you can see that there is, in fact, here a frame. There's kind of a golden cow there, and it looks like people around it are worshiping that cow. And it was a little bit more complicated by even people who were theoretically well-versed, that is J.M. Peebles, AM, MD, and PhD, had complained of vaccination as a curse and a menace to personal liberty. So the more things change, the more they stay the same. Now, as I mentioned, the lady, the milkmaid, who was responsible for having cowpox used as a vaccine was named, but also was the cow. And the cow was named Blossom. And here's the hide of Blossom, which is kept at St. George's Hospital. And so this is a picture of St. George's Hospital as it was probably about that time, right across Hyde Park. Unfortunately, this building was sold to a group, and it is now a hotel. And so the hide of Blossom is in Tooting in London, in the suburbs of London. Variolation continued in parallel with the smallpox until many years after Jenner's death. But nevertheless, it was one of the more successful vaccines there ever was. Here is smallpox eradication by date. In America, it was eliminated by 1952. In the Western Hemisphere, completely by 1971, including variola major and variola minor. And then the last case of variola major was in 1975 in Bangladesh, and in 1977 of variola minor in Somalia. And here is the man who was the last known case of smallpox. And at that time, if I remember, there was a bounty of $10,000, maybe, which is a lot of money in those days, for anybody who identified a case. And so there were lots of people making sure that smallpox really had been eliminated. He worked in a hospital, not sure how he got the variola minor, but he made a full recovery. And unfortunately, he died of malaria, a disease for which we don't have a vaccine yet. Many people have been working on it. Now, he wasn't the last case, the last human who developed smallpox, because in 1978, a photographer, illustrator working at the University of Birmingham became ill and died of smallpox. She had become infected through inadequate ventilatory exhaust of the laboratory below. Now, you wouldn't blame the poor guy who was a well-known virologist for the engineering of the laboratory. But as they started looking at this, it turned out that there were a lot of problems with that laboratory. They were not following procedure. And Dr. Benson was clearly going to have a bad ending to his career, which he obviated by committing suicide by cutting his own throat in a garden shed outside his home. And subsequent to that, the world decided that there was no point ever in doing any experiments with smallpox, and the stocks that were available in laboratories were destroyed. Although we all know that our armed forces and those of other countries, including Russia, have plenty of stocks that can be weaponized at any point. Now, lest you think that things are over, this was just published in the MMWR earlier this month. And this is the case of a man who came to a hospital in Dallas with monkeypox. And he did well, although he was in the hospital for quite a while. So it wasn't just a benign illness, it was a serious illness. And he, in addition to having arrived and having to come to Dallas ED and been hospitalized for quite a bit of time, as you can see, a month, quite a bit, he also exposed an enormous number of people, 147 contacts in the flights, 4 in the hospital. And then the laboratory sent some samples to Utah where they were not handled very effectively. And so there were another 46 people there who had to be dealt with. And presumably they were all inoculated, as people of a certain age like mine have already been. Although this is not a lifelong protection as far as I can tell. And I can tell because I was vaccinated a few times. So much for smallpox. I think the next big increment was when the rabies vaccine was developed, and rabies have been known about for at least 4,000 years. And in fact, the phrase dog days reflects to the summer when the brightness of the dog star Sirius. And during the dog days, the dogs were believed to be more susceptible to the madness that we now call rabies. And here's a prescription that comes out of this article by Miller and Nathanson. In the Talmudic prescription was you write prayers on the skin of a male hyena or leopard. I don't know where you find that. You strip off your clothes, you bury them and burn them after 1 year. You drink only from a copper tube so as not to see the shadow of the demon. And although I suspect this did not work for rabies, the truth is that we really don't have any better treatment now anyway, since we have a good vaccine and the vaccine is good post-exposure, but once someone has rabies, it is almost uniformly fatal. Although some people are put in a coma, and that seems to help sometimes. Here's Louis Pasteur, a drawing with his rabbits, and the rabbits were critical to Pasteur's experiments, and particularly his experiments with rabies. But that's not where he started out with. He started out working with vaccines against bacteria. And in fact, he was working with cholera and anthrax, and he had an assistant who was to inoculate chickens with the fowl cholera. But the assistant kind of forgot about it, and he left the cultures lying around. And then when he remembered, he decided, okay, let me just go ahead and inoculate the chickens. But at that point, the bacteria had been attenuated and the chickens never got very sick. Now, Pasteur may have had a chance, but he was also a very bright man. And he looked at this and said, let me try and find out if I can re-challenge them with fresh cultures. And when he did that, he discovered that, in fact, the cholera that had been lying around and now learned as a vaccine. And he is, in fact, the first individual who coined the term vaccines in honor of Jenner, who, of course, he knew about. And this sort of goes to the old saying that chance favors a prepared mind, because it was pure chance, but he was ready for it. And this is his description of the vaccine, and I'm sorry it's a little bit fuzzy, but it was in 1881. And I'll just point out the one sentence here: the subject of my communication is inoculation in relation to chicken cholera and splenic fever, and the statement of the method for which we have arrived at these results, a method the fruitfulness of which inspires me with boundless anticipation. I'm not sure if this was a translation or his own writing in English, but English is clearly the language of science, and it has been so for quite some time. So, there is some thought that rabies had already been transmitted into rabbits by taking rabbit brains and sticking it right there. And by other investigators. This point is controversial. Some people say that it was Pasteur who first did this, but what he was looking for was a short, easily studied system. And so what he did was he passed it brain to brain with the goal of shortening the incubation period in rabbits and to have a better model. And this, by the way, is a classic rhabdovirus particle, or rabies virus. It's bullet shaped, that's where the name comes from. And you can see the glycoproteins kind of sticking out of the lipid bilayer of an envelope virus, and inside this dense core of the RNA with some proteins that are necessary for transcription, immediate transcription. Anyway, he then started deliberately going to attenuate the rabies. First, he did the shortening with rabbits, and he also took the spinal cord and dried it. And then he tried to immunize dogs. And of course, dog rabies was a huge problem then, as it is in other parts of the world, not so much in ours. And he called this attenuated rabbit isolate fixed, as well as opposed to the original isolate, which he called the street virus. And that terminology actually continued for many years. I guess word got around, and in 1885, this 9-year-old boy, Joseph Meister, this is actually a little later than that, it's 1989, so it was 4 years later, he was brought by his mother from Alsace to Paris, where Pasteur worked, and he had been bitten by a rabid dog, and somehow the word had gotten out that he was working on this vaccine. And so Pasteur kind of reluctantly, and I would say bravely, added that part, he inoculated him 14 times with the rabbit's spinal cord that had been progressively air dried for shorter periods, so that the preparations were less attenuated, and he survived. And since then, as it is now, rabies is considered a fatal disease, the fact that he survived was sufficient evidence that the vaccine had worked. Obviously, in this case, it would have been really brave to go ahead and inoculate him with rabies, and he obviously didn't do that. By 1886, or 1 year later, he had already treated 350 patients and was famous for having doing that. Now, this young boy then became a man and was healthy, and he worked at the Pasteur Institute, doing all kinds of things. And after the occupation of Paris by the Nazis, he committed suicide. It wasn't clear whether he was upset about what was going to happen to the Institute or what was going to happen to his family, whom he could not have any contact with. He stayed in the Institute after the Nazis occupied the city. But here's the original description, and I won't bother reading the French since you would all be appalled at my accent, but it says a method to prevent rabies after biting. Now, as we know, it is one of the few diseases where you can have, well, not only one, but you can have effective vaccination after exposure. And the reason for that is that the incubation period is very long. The reason the incubation period is very long, which Pasteur could not have known, is that the virus does not cause a huge viremia, or if it does, it's not sufficient to enter the brain or the central nervous system, but rather it spreads into the brain and central nervous system by retrograde axonal transport. And depending on the length of the nerve, that could take quite a long time. And so someone who is bitten in the legs, and particularly if it's an adult or a tall adult, is going to have a much longer incubation period than someone who is bit in the face. In the United States, we have essentially no dog rabies. All of our animals are inoculated, but we still have occasional cases of rabies, mostly transmitted by bats, and those usually affect the face. So the amount of time available for post-exposure prophylaxis is limited. But fortunately, there are very, very few cases in the country. I didn't look at the number, but it's probably one in the handful in the last 10 years, if any. Now, Pasteur, of course, now famous for that. And here's the centenary, the 100-year celebration of his of the vaccination in September of 1985. But after the vaccine first came out, it wasn't uniformly accepted. And here is a paper in 1987 first read at the New York Clinical Society by Herman Briggs, and where he says he has been somewhat misunderstood. It was desired to correct this erroneous impression that finally overcame my scruples. I have been wrongly regarded as an enthusiastic supporter of and believer in Pasteur's method for the prevention of rabies. And he goes on and says that he really doesn't quite believe in it. I didn't bother reading through his logic. It is true that after Pasteur actually was very finicky about his notes, and he told his family he didn't want his laboratory notes publicized, and they weren't for many years. When they finally were, how that first human inoculation, where it came from, was not entirely clear, as where some of the data were also a bit on the unclear side. Now, one of the things that's pretty obvious is there was never a real trial in the matter that we would now consider scientifically sound. Basically, the vaccine was given, the child didn't die. That happened on a number of occasions, and on. And of course, that's what we do now. And so we're pretty sure that it works, but we never had a proper trial, and no one is ever going to do that, because who's going to do post-exposure prophylaxis with a placebo? It would be horribly inhumane and inappropriate. And so that's where we are. Now, his method was eventually changed. A simple vaccine was a chemical inactivation, but unfortunately, because it was coming from nervous tissue, some individuals were presumably immunized against it and developed a form of experimental allergic encephalomyelitis, a well-known animal model for MS, until eventually, and this was rare, but it did happen, and then eventually the virus was produced in duck embryos, which still has some problems, then mouse brain, and then diploid cell lines such as WI38s and MRC5s. And there are still some various preparations, but rabies vaccines are now all done in tissue culture. And this issue of EAE, or neuritis as it was called at the time, is no longer a problem. Now, rabies has been eliminated from our country, but it still is out there. And in some countries, there's about 60,000 worldwide deaths per year. This is data from 2017, and no reason to think that it has decreased. A lot of it is from dog exposure, but there are other animal exposures as well. In the Western Hemisphere, it's less than in Asia, for example, and it comes up all the time. My looking at this map would tell me that this is Ukraine with the Crimean Peninsula here and the Sea of Aziz right here, and it's got some possibility for rabies, and it is listed by the CDC as a country where there is rabies. And you say, why is he bringing this up? There have been, as we all know, millions of people leaving Ukraine in the last 2 months since the Russian invasion, and naturally they bring their dogs. And one of the problems that they have run into is that getting their dogs into the United States is a bit of a problem, and it is as well into some parts of Europe. Although I read in the Times a few days ago that even though Mexico is here listed under 4.5, Mexico is okay. So one route that people were taking is going to Mexico and sneaking in by having Americans bring the dogs in as if they're the owners. And so now there's a workaround for the fact that that you can bring dogs in from the Ukraine, and hopefully that will be fixed soon. Now we're going to change topics and go to yellow fever. And yellow fever, again, has been known for quite some time. This is a pamphlet on yellow fever of Jamaica, common in the tropics because, as we now know, but wasn't known for many years, it is transmitted by mosquitoes, and specifically by mosquitoes that live in the tropics. You may never have thought about this, but obviously not all mosquitoes. We have mosquitoes here. We have mosquitoes in Wisconsin where it's very cold, but the species of mosquitoes in Wisconsin can overwinter by simply laying dormant in tree holes, whereas the species that transmit yellow fever cannot do that. So they're really tropical mosquitoes. And the relationship between a virus and a mosquito is usually a biological relationship, not a mechanical relationship. So the virus really has to replicate in the mosquito. Anyway, it has been known for a long time But after the experiments of Walter Reed, and we'll get back to that, it was known that it was transmitted by a mosquito, and this is from the National Library of Medicine. Looks like it's the 1930s or 40s, and this is talking about destroying their foxholes and the mosquitoes, because yellow fever has been a real problem anytime there are massive groups of people working in the tropics, such as when the Panama Canal was built. Yellow fever has a huge history also in our country. This is a drawing depicting a massive epidemic in Philadelphia in 1793, where volunteers were going around picking up dead bodies to bury them. At that point, of course, it wasn't known that it was transmitted by mosquitoes, so people were afraid of touching bodies or anybody who was sick. The people who lived in Philadelphia were essentially quarantined, and they couldn't leave the city at the risk of getting kicked back or even shot by the suburban people. And you may remember that Philadelphia was the capital of the country at that point. Washington did not exist, Washington, D.C. Washington lived in Philadelphia. He had a house in what is now called Independence Mall. His house foundations have been discovered. And I guess I have enough time to digress for a bit. As we all know, the father of our country was an enslaver, and he brought some of the enslaved people to work for him at his house in Philadelphia, but he rotated them because at the time the rule in Pennsylvania was after a certain period of time, if a slave or enslaved person was in Pennsylvania, he or she became automatically free. So he actually went through the trouble of rotating them so that none of them would stay in Pennsylvania long enough to be free. Be as it may, he and the rest of the government had to leave Philadelphia and go up to Chestnut Hill, up in the hills, which is still part of Philadelphia now, because the city was in terrible shape, and they obviously didn't want the whole government to be sick. Now, yellow fever is a flavivirus. Flavy is the Latin word for yellow, and it comes from the fact that you have jaundice. The yellow jack was a banner that was used in an area that was quarantined, particularly ships, and it became a particular concern in 1898 and the construction of the Panama Canal. It was about that time that it was determined that it was not a bacteria and that it was really transmitted by mosquitoes, but there was no effective laboratory with which to experiment. At the Rockefeller Institute, they thought maybe it had been a spirochete and it was produced, but obviously that didn't do much. And so eventually an African strain from a Ghanaian man named Nasibi was transmitted to rhesus macaques, which is an Asian species. Presumably the African macaques would not be susceptible. And then Max Tyler, a famous virologist, and his colleagues succeeded in developing a mouse passage model, which attenuated the strain for the liver and developed some neurotropism. It was administered with immune serum to make sure that there was no residual active virus. But then some people still got jaundice, probably because they got hepatitis A or maybe B from the serum. The French vaccine was done a little bit differently, and that actually continued for quite some time. Eventually, they grew the virus in embryonated eggs and not in tissue culture. It's still in eggs as influenza. And the 17D and its derivatives is still used as a vaccine. It was licensed in 1938, and it provides lifelong immunity with one inoculation. Here's a picture of Max Tyler. He received the Nobel Prize in 1951 for developing the yellow fever vaccine. So far, the only prize that has been given for directly developing a vaccine. He was South African. And so he was the first African Nobel laureate, but he did most of his work at the Rockefeller Institute. So we'll move on to polio, and I think we're doing okay time-wise. And, at the peak in 1944, polio had 19,000 cases, and this is cases of paralysis for somebody not of just GI disease, with 1,400 deaths. And it was an important cause for philanthropy, and it was supported particularly by FDR, since, as we all know, he had polio himself as an adult. And there was a National Foundation for Infantile Paralysis. Every year, they celebrated his birthday, and it was called the President's Birthday Ball, to raise money. And it was Eddie Cantor, a comedian, who is credited with the idea that he said we should send a March of Dimes. I guess in those days, dimes were more valuable than they are now. And the name of the March of Dimes for the foundation is what stuck. And of course, the March of Dimes still exists, although they now concentrate on other childhood issues and no longer in polio, which has not been 100% eliminated, but almost 100% eliminated from our country. Now, the guy who is credited, who received the Nobel Prize for polio, wasn't the one who created the vaccine. We all know his name is Jonas Salk, but it was a man named John Franklin Enders. And what he did is he developed a methodology for culturing the virus. And he had an interesting history. He majored in English literature, worked in his family's real estate business, I think it was, and didn't particularly like it. So he eventually went to Harvard, and he originally graduated from Yale, it's important to mention. And he eventually went to Harvard and got a PhD. And then he went to work at the Boston Children's Hospital. Other people were working on polio. It's a big deal. And he was kind of a junior guy, and he sort of said, well, just work on mumps or something else. And he did. And one of the experiments that he was doing was trying to grow the chickenpox virus in tissue culture. And this case, tissue culture, were really from human embryos. Nobody talks about where those human embryos came from. Growing chickenpox, by the way, is really hard. And so, not surprisingly, I don't think he was very successful at doing that, but they did develop a methodology for doing tissue culture. They put the growth factors in, they knew how to change the media, they added antibiotics to prevent contamination, something that you no longer have to do now, but we have better mechanical ways of preventing that. And then one day they had some leftover tissue and they added some of the Lansing strain of polio, which had been grown in brain. And sure enough, they were able to culture. And here's the paper in 1952. And what we're looking at here is the days after inoculation of the tissue culture. And then on the y-axis, we're looking at the LD50s in mice. And as you can see, they use all kinds of different tissue from a single human embryo: intestine, skin, muscle, adrenal, brain, heart, spleen, lung, liver. And all of them gave some result. Not all, well, not all of them, actually not all of them, but most of them gave some result. But particularly the skin, muscle, and the kidney were very fruitful places for growing the virus. And so growing the virus here in tissue culture was what first became the model for developing the vaccine. He himself or his laboratory did not work on it, although some of the folks in his laboratory wanted to, but they were kind of dissuaded from doing that by him, because he felt only pharmaceutical companies should be doing that. But he was still credited with the growth of tissue culture. He was nominated for the Nobel Prize on 2 occasions. And the lore is that he didn't want to get the prize unless his collaborators shared with him. And this is from the Nobel Prize citation. And I won't read the whole thing, but in 1941, Frederick Roberts and Thomas Weller, all 3 of them, succeeded in culturing the virus that causes polio in human muscle and tissue in a laboratory setting. And that's why he was given the Nobel Prize. He actually went on to figure out how to grow measles virus, and he formed the basis for the development of the measles vaccine. And he was on the cover of Time magazine in 1961. He is virologist John Enders. And then if you're looking carefully, you can see that the issue cost 25 cents, and you could get a whole year worth of Time magazine for $7. Okay, so Salk actually may have actually gotten some of the cultures from Enders, but he started he produced a vaccine that was inactivated and was tested on 1.5+ children in the US, Canada, and Finland. And in 1955, the vaccine was adopted in the US, and the incidence dropped dramatically, more than tenfold initially, and probably beyond that. It wasn't clear what strains were going to be used, and that became the standard. And this is in the UK, and you can see the graph. The introduction was a little bit later, 1956. You can see very quickly how polio disappeared how effective that vaccine was. But there was still a lot of argument about the use of inactivated versus activated versus attenuated vaccines, live attenuated vaccines. And there was one incident that occurred in 1955, published here in 1963. In those days, especially the publish wasn't as strict, and after all, Neil and Alexander Langmuir were at the CDC, where they didn't have a huge pressure. But they described the Cutter incident. It was that there was a laboratory in Oakland, as I recall, that did not do the proper inactivation. And so it actually caused polio in a number of children, 60 to 80, I think was the number. And that, of course, revived the issue that it was a mistake to use an inactivated vaccine, that you really needed to use an attenuated vaccine because the inactivation you could never count on. Of course, people forgetting that it was only one isolated instance in one laboratory that just wasn't following protocol. But nevertheless, it continued that debate. And then attenuated vaccines were produced by many, but particularly Hilary Kaprowski at the Wistar Institute, Cox at Letterly, and Albert Sabin, which is the one that we know of at the Children's Hospital of Cincinnati. And it was his vaccine that's eventually accepted in 1961-63. It was easy to produce, a herd effect, because the children would excrete the virus in their diapers, and the parents would then get re-inoculated. But the problem was that reversion was very fast, particularly types 2 and 3, and there were 3 types inside. And that was a problem for many years, and became a bigger problem as the population aged, 1, and 2, there were more people around who had transplants, chemotherapy, and all kinds of things that attenuated their immune system. And so at this point in the United States, and has been that way for the last 22 years, we only use the inactivated polio vaccine, although other countries, because of the ease of administration, still use the attenuated polio vaccine. Now, and this is I won't go through it because of time, but just to show you how each of these 3 times it was really done, you make it up as you go along. First, they pass it in monkeys, then they go in kidney cells and monkey cells, then the plaque purification, which is to make sure that it's a single species, something that now we would do molecularly. And so these vaccines, this is exactly how they weren't even done exactly the same way each time. And these 2 are particularly prone to reversion into a virulent phenotype, and remarkably, well, we won't have time for it, but there's many changes between the original and the attenuated vaccine, but only a few changes are necessary for neurovirulence to return. Now, I bring up Hilary Kaprowski because this vaccine was used in Africa, and now called in areas called the Democratic Republic of Congo, Rwanda, and Burundi. It was apparently was good, but it was never approved for the US. And part of it had to do with the politics. None of these people had small egos. Sabin was reputedly not a very nice man by anybody who reads about it. And my late uncle, trained in Cincinnati, told me he was really quite nasty. Hillary, on the other hand, was quite charming, could be quite charming, as well as irascible. And I actually saw both sides of him at different points in my life. He was a terrific pianist, always had a concert at the Wistar Institute. I heard him once in the house after we had dinner, in the house of a famous neuropathologist. And his vaccine disappeared, and you would have thought that, why am I bringing this up? Only because I know him? No, because it turned out that in 1990, it was already known that AIDS had been a virus that had jumped species from chimpanzees to humans. So this guy, Tom Curtis, decided that he had the answer to how AIDS had been crossed into humans, and he said that it had really been Hillary's fault. And his reasoning was that Hillary had grown the poliovirus in monkey kidney cells, and that in doing so, he had used chimpanzees, and those chimpanzees had the chimpanzee SIV, CPZ as it is called, and it was that jump that he then inoculated, that preparation that he then inoculated into thousands of people in the Congo that led to their infection with this SIV, CPZ, and ultimately to AIDS. And of course, published in Rolling Stone. Lots of people talked about it. There was a huge outcry. Hillary said, no, 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 I didn't do that. The monkeys were macaque monkeys. Macaque monkeys come from Asia, not from Africa. There were no chimpanzees. And it died down for a while. But then Edward Hooper wrote a whole book about this, where he wrote about the origin of HIV and AIDS being the Hillary vaccine. Fortunately, the original stocks were kept at the Wistar Institute and in London. There were 2 sites, and they were pulled out. 3 different laboratories looked. They looked for 2 things. First, for any evidence of HIV in them, there was none. Any evidence of chimpanzee DNA, there was none. And so eventually, after a lot of work and many different people, and then there were some other genetic stories that I don't have time to go into, everybody agreed this is complete bonkers, and it's one of those crazy theories. But believe it or not, this still gets brought up by the anti-vaxxers because they don't believe that the laboratories didn't find the chimpanzee virus. So I have 2 more vaccines to talk about. One of them is hepatitis. And again, here is Baruch Blumberg, commonly known as Barry Blumberg, who received the Nobel Prize for the discovery of the virus that causes hepatitis B. He was really looking at polymorphisms in proteins, and so he had developed good ways of looking at proteins. And he found that in the indigenous people of Australia, there was this unusual protein that kept propping up, and he called it the Australia antigen. And then he and his collaborators discovered that this antigen seemed to appear in individuals who received lots of transfusions. And so they began to test the theory that this was potentially a signal of a virus. And by the way, to this day, hepatitis B and the specific strains are very endemic in the indigenous population of Australia. Not exactly clear why, and I'm not a hepatologist, so I don't know how that carefully, but it still continues to be a problem. And so he discovered the Australia antigen, which we now know is the surface protein of hepatitis B, and pretty quickly began to associate it with transfusions. This is an interesting abstract published in 1971, which I brought up because here's 2 hospitals. One is the Philadelphia General Hospital, and the other one, the Hospital of the University of Pennsylvania. These are literally across the street from, or across, well, no, not even a street. There's an alley, across an alley from each other. And they had huge differences in the amount of transfusion-associated hepatitis. Very limited at HUP and very common at PGH. And so John Senior, who's not here, but he was the senior author, named is Senior. And he and his collaborators started screening the blood for the Australia antigen, and very effectively demonstrated that if you excluded blood samples that had the Australia antigen from transfusion, the incidence of hepatitis was quickly diminished. Dr. Blumberg, and I'm going to skip this slide, just briefly mention that he then started experimenting with the idea of a vaccine against hepatitis. And so at this point, this is in the Nobel laureate lecture given in 1976. He had already now called it hepatitis B surface antigen, and he had inoculated patients who were going to undergo dialysis, and those who entered without antibody had high probability of developing the antigen, and those who did not after several months of dialysis. And so there was actually a vaccine that was used as an extract of the virus. People were a little reluctant about it because of the possibility that it might transmit other viruses, until Genentech in California and company managed to do one of the first serious cloning experiments in 1982, where they were able to clone hepatitis surface B antigen, the surface protein, synthesizes in yeast, and then be able to make this into particles that had properties that were similar to the 22 nanometer particles that are present, secreted from human cells that are infected with hepatitis B. And that, of course, is the way that we now all get immunized. There are actually 3, at least in this country, that use the same process, same basic idea. I think I received with CombiVax, and the difference is the adjuvants that are used. I am unfortunately not going to talk about adjuvants because we don't have time. I want to give the last few slides, but a huge advance has also been in understanding how adjuvants work and how you can tailor adjuvants to specific arms of the immune system so that you can end up with an effective vaccine. Let's go to the RNA vaccines in the last few slides. And the first thing to say is, you would think, why didn't they do this before, right? I mean, you think RNA makes protein. Why didn't somebody come in and throw some protein in and some RNA in and watch the protein being made and see what happens? Well, it's a lot easier said than done, like many things. First of all, there are like 20 different spots where RNA is bound to cause trouble. First of all, it gets chipped off by ribonucleases, and then it gets recognized by all kinds of systems in the cells, and they then in turn lead to interferon production as well as a variety of other cytokine productions, and that can lead to a huge cytokine storm and deaths. And so a lot of the problems that the people working on this had were, first, that the RNA never made it to the cells to be made into protein, and 2, when you inoculate huge amounts of it, what you would end up with is a dead experimental animal because of the cytokine storm that would occur. And so the big change was actually, in retrospect, although they tried many things before it came to this, was really stabilizing the RNA. And this is a work for which Carrico and Weizmann have now become quite famous and received the Lasker Prize last year, and I'm sure they're on the short list for the Nobel Prize. And what they did is they really flipped the amino and the carboxyl group. And so you end up with this pseudouridine, which has the same base pairing properties as the original uridine, but is much more stable, lasts much longer in the circulation, and it lasts much longer in cells. And then there was one further modification added here, which is from uridine, you go to pseudouridine, which is what I just showed you, but then you add a methyl group, and that makes it even more stable. It's not toxic. It is still translated. It does not get integrated. And so it is a very effective way of making RNA vaccines. The second part is how to deliver them into the cell. And this is from the pharmaceutical company called Acuitas. And I believe that's the one that we have, those of us who have received RNA vaccines, have received the liquid, so lipid nanoparticles that are made by them. They make the nanoparticles that then package the RNA, and that little gemisch is what we end up getting, goes into our cells. And there's one other change. This comes from respiratory syncytial virus, and without going into great length, is the trimeric glycoproteins of RSV and of SARS-CoV-2 undergo a change that they need to have in order to penetrate cells, either at the cell membrane or at the endosome. And the reason is that they need to expose hydrophobic amino acids that are usually buried within the glycoprotein to interact with the lipid bilayers and therefore discharge the contents of the virus into the cytoplasm. With RSV, it was shown, after a huge problem with the original vaccine, that the epitopes that are important for neutralizing the virus, which are listed here in red and orange, disappear in the fusion form of the protein. And the virus goes back and forth between these two. And so the children who were vaccinated with this preparation actually got worse because they did not have any neutralizing activity, but they had antibodies sticking to the virus, which then allowed the FC portion to go into dendritic cells and get the virus to infect since it had not been neutralized. SARS-CoV-2 does essentially the same thing, although the important sites are not as well defined here. And so the other big trick that occurred from Barney Graham at the NIH, although the companies won't tell you whether they used his idea or their own idea is to stabilize the protein in this conformation by adding 1 or 2 prolines at a critical point. And so the vaccine that we get, the RNA vaccine that we get, has fake RNA. That is, it has a modified form. It is put into lipid nanoparticles so that it can be programmed to put into cells. And 3, it has an alteration in the protein so that it does not turn into its post-fusion spike, and it allows us to make antibodies against it. And so this is how it works. First, you have the spike gene. It's made into RNA vaccine, put into this lipid coating, put in. The viral spike is made. A couple of ways to do it. Here's a vaccinated cell. The viral spike is expressed. The B cell recognizes it, makes short-lived plasma cells that make antibody. The antigen-presenting cell, the dendritic cells, can digest the proteins. They have helper T cells, which end up making more T cells, or the long-lived plasma cells that are responsible for antibody for a long period of time. And this is how we ended up with where we are. And by the way, this is Carrico and Weissman getting their vaccine just like everybody else. And although they originally owned the patent, that patent was sold, and so they're not getting the millions. Those are going to a patent troll that bought the patent from the Penn Medicine. That's another story of a huge mess. So I tried to think of a summary and I couldn't think of one. So this is what I because there's so many different things that are going on, and all I said, well, it's just like everything else human. There's serendipity and chance, there's tenacity and resilience, there's adaptability, there's inventiveness, but the biggest stimulus for the development of a vaccine is an external threat. And I think I'm only 5 minutes over time. So thank you all.
Thank you so much for that incredibly fascinating talk. You have had quite an illustrious career and have really witnessed a lot of dramatic change over this period yourself, I imagine, not the least of which we've all seen in the last couple of years with this huge advancement. Does anybody have any questions? I have a couple, but I want to offer up to the audience to ask first. I'm not hearing anyone or seeing anything in the chat box. So let me ask you, with this new RNA technology, what do you see in the future? How else can this be implemented in other infections?
Well, I mean, you can let your imagination run wild on this. There are now many versions of the SARS-CoV-2 vaccine that are being made. Some presumably are for the variants. Some just include the receptor binding domain. Some don't have the transmembrane domain, so they free float. And I'm sure other viruses will be looked at that as well. But once you can deliver RNA into cells so effectively, you can look at vaccines against cancer. You could look at some potential correction of some genetic defects. Obviously, not all will be susceptible to this, but some will, particularly if it's an issue of that you can correct it by a gain of function. And so, I mean, they're already in SMA, for example, there are things that you can do to manipulate, just express the right protein. And that's actually the way also that sickle cell is being potentially treated, is by expressing fetal hemoglobin, I think is how it is, if I remember right. So you can immediately think of how all of these things can be done very effectively with RNA vaccine. There's a whole industry that is now based on doing this. And again, I'm sure there are 1,000 ideas, of which quite a few will work and some will not.
Mm-hmm. So
it's also very simple to make. It's also very simple to make, and because when you make a protein vaccine like Shingrix, for example, or the hepatitis B, you need to have your own purification method for each of the proteins, right? You can't use the same purification method for the Shingrix vaccine that you use for the hepatitis B surface antigen. They're different proteins. They're going to behave differently. They're different sizes. So each one has to have its own purification method. requiring a lot of experimental work. For the RNA vaccines, all you need is the RNA. So if it's going to work, the RNA is purified the same way. It's just the sequence is different. So there's a huge potential advantage for things, particularly that may not have a huge market, let's say, but it's not a huge investment in time either. So
sounds like a lot of very exciting potential. I guess my other question, going in the opposite direction, is going back to the earliest history of vaccine development with smallpox and that history of an anti-vaccine sentiment even hundreds of years ago. Is there anything learned in all of these I guess controversies created by human, less so science?
That's above my pay grade. I don't know what to say. I think I brought it up because just to say that humans are humans, and conspiracies are conspiracies, and scientific skepticism is common, and it was common in the 18th century. I'm sure it was common before then too. It just we don't have a lot of data before then. And it will continue to be common, and there's always going to be a minority that just don't believe in the science. I don't know. I mean, I guess my only answer would be I wish we educated people better at an earlier age. That would be my answer. Better education of the children on the importance of scientific principles, and I don't think we do that very well in this country. But there are a lot of factors that are against that.
Well, lots more work to do. So thank you so much for this
My
incredibly
pleasure.
fascinating.
Take care.
I enjoyed it. Take care.
Bye.
Thanks, everybody.